In this topic, we're going to look at the structure and functions of blood. So we're going to look at what is blood and the different constituents: blood plasma, red blood cells, white blood cells, and platelets. So what is blood? Well, blood is the medium by which materials are transported between different parts of the body. So it's made up of plasma, and then your two types of cells, red blood cells and white blood cells. So if you have a look at the test tube on the right, you can see the different composition of blood. So plasma makes up about 54% of the blood, and then your white blood cells and red blood cells together make about 45 to 46% of the blood. So what is blood plasma? Well, blood plasma is about 90% water that contains only about 10% chemicals that are dissolved or suspended in it. And the function of the plasma is to transport these chemicals along with heat from where they are produced or absorbed to the cells that use or excrete them. Now, plasma can be forced out of the capillaries into the surrounding tissue. We then call it tissue fluid. So it doesn't have plasma proteins. Tissue fluid doesn't have plasma proteins. So let's have a look at the different chemicals that are transported in plasma. You've got nutrients, for example, glucose, amino acids, and vitamins. Waste products, for example, urea. Mineral salts, for example, calcium and iron. Hormones, for example, insulin and adrenaline. Plasma proteins, for example, albumin and fibrinogen. And then finally your respiratory gases, oxygen and carbon dioxide. So between them, these chemicals make the plasma slightly alkaline, about pH 7.4. Now the removal from the plasma of the proteins that are involved in clotting, for example, prothrombin and fibrinogen, results in a liquid called serum, which does not clot. Okay, let's have a look at red blood cells. So these are also called erythrocytes, and they have biconcave discs. This means that they are dented in the middle like a donut. They're about 7 to 8 micrometers in diameter, and they're very narrow. There are about 5 million red blood cells per cubic millimeter of blood. Now, red blood cells only live for about 120 days or 3 months, so this means that they need to be constantly made. So they're made in the bone marrow of the sternum, skull, ribs, pelvis, and vertebra. They then get broken down in the spleen, which we'll have a look at in a little bit. So let's have a look at the different features of red blood cells and their functions. So red blood cells are unusual as they don't have a nucleus, mitochondria, rough endoplasmic reticulum, or Golgi apparatus. So this increases the volume of hemoglobin that can carry oxygen. They're thinner in the middle, so they've got a biconcave shape, so that there's a larger surface area to volume ratio for oxygen exchange. They have a flexible membrane that easily changes shape, so that the red blood cell can flatten against a capillary wall, and this reduces the diffusion distance for gaseous exchange. They contain a red pigment called hemoglobin that carries oxygen. So as I mentioned earlier, the red blood cells last about 3 months, and then they're destroyed in the spleen. So they're broken down into globin, which is the protein, and the heme group. So the globin is broken down into amino acids, and then the iron is removed from the heme group, and this iron is attached to proteins to form transferrin. The rest of the heme group is used to make bilirubin, which is used to make bile. Okay, white blood cells. So these are also called leukocytes, and the spelling of leukocytes, sometimes with a K, sometimes with a C. So as you can see here, they all contain a nucleus, and they're either spherical or irregular in shape. They are larger than red blood cells, and they're about 7,000 white blood cells per cubic millimeter of blood. They're made in the bone marrow of limbs, and they're involved in the immune system. Okay, what are the different types of white blood cells? Well, white blood cells can be divided into two groups based upon how they carry out their function to protect the body against infection. You've got phagocytes and lymphocytes. So phagocytes remove microorganisms and dead material by a process called phagocytosis. How to recognize them? They've got a lobed nuclei. Lymphocytes, on the other hand, secrete antibodies that immobilize and kill microorganisms and make them ready for phagocytes to engulf. So they're quite specific. How to recognize them? They have a large, round nucleus and a small amount of cytoplasm. So monocytes and neutrophils are phagocytes, and monocytes are the largest white blood cells, so they can develop into macrophages. Both monocytes and neutrophils have got these lobe-shaped nuclei, whilst lymphocytes have got a large round nuclei with little cytoplasm around them. So phagocytes engulf bacteria by phagocytosis. The macrophages are stationary in organs, whilst neutrophils are found in the blood and lymph. Then you've got your lymphocytes. So here you can see the lymphocytes have got that large round nucleus that dominates the cell. So there's little cytoplasm around it. And you find lymphocytes in the blood and lymph. White blood cells are divided into two groups based upon how they carry out their function to protect the body against infection. So let's have a look at the difference between phagocytes and lymphocytes. Looking at the structure, remember that phagocytes have got these lobed nuclei. Lymphocytes have got a large round nuclei and little cytoplasm around them. The number, phagocytes, they're about 4,900 per cubic millimeter of blood. Lymphocytes, 1,680 per cubic millimeter of blood. Examples include macrophages and neutrophils for phagocytes. Lymphocytes, well, you've got the special lymphocytes, B cells, T helper cells, and T killer cells. So we'll look at that in a lot more detail when we discuss immunity. Where they're found? Well, phagocytes, the neutrophils are found in the blood and lymph, and the macrophages are found stationary in the organs. And your lymphocytes are found in the blood and lymph. Their functions, well, phagocytes are involved in phagocytosis, and lymphocytes are involved in the secretion of antibodies and cytokinins. And where they're manufactured, well, phagocytes are manufactured in the bone marrow, and lymphocytes are also made in the bone marrow, but they mature in the spleen or the thymus gland. Sometimes white blood cells are differentiated according to whether their cytoplasm appears granular when stained under a microscope. So looking at granulocytes, these have got lobed nuclei, as you can see here, and they include neutrophils, eosinophils, and basophils. So their cytoplasm appears like little granules. It has little granules in it. And then agranulocytes include lymphocytes and monocytes. Just quickly, basophils make histamine and heparin, and eosinophils have antihistamine properties. Finally, what are platelets? Well, there are numerous platelets in the blood. There's about 250,000 per cubic millimeter. So these are membrane-bound fragments that are formed from megakaryocytic cells that were made in the bone marrow cells. They're involved in initiating blood clotting. So let's have a look at what this blood clotting mechanism involves. Now, when blood vessels are damaged, the platelets come into contact with that tissue, especially the collagen, and these platelets break open and release serotonin. This causes the smooth muscle in the arterioles to contract and narrow the vessels. Thromboplastin is an enzyme that converts, in the presence of calcium ions, prothrombin to thrombin. Thrombin converts soluble fibrinogen to insoluble fibrin. And then fibrin forms a net over the damaged area. So the red blood cells and platelets are caught in this fibrin mesh. Contractile proteins in the platelets contract, making the clot tighter. Right, in summary, what have we looked at? Well, we discussed the different components of blood plasma, red blood cells, white blood cells, and platelets. So we looked at blood plasma and the molecules that are carried in it. We also looked at red blood cells, where they are made, where they are broken down and what they're broken down into. And then we also discussed their different features that make them good at carrying oxygen. We looked at white blood cells and how they are classified into phagocytes and lymphocytes, and how to differentiate them from each other. So remember that phagocytes are involved in phagocytosis, and lymphocytes are involved in secreting antibodies and cytokinins. And then finally, we looked at platelets and how they are involved in blood clotting. And that concludes our lesson. The end.